Thanks, Doro, for the very kind introduction and for also connecting my work so nicely to the interests of this community here, working in autophagy, mitophagy, and related fields. So it's really truly exciting to be given the opportunity to present to you all, and I am really grateful for that. So as Doro already said, we use theoretical methods, and primarily we use molecular simulations to study molecular processes in living systems in an effort to understand cellular functions, and increasingly going from molecules all the way up to cells. And so molecular simulations, I think, and I hope to convince you in the following half hour or so, are really promising a solution for this structure to dynamics to function challenge, because they give us a physics-based representation of the molecular system that's transferable, and we call this a force field. And then they allow us to conduct, in effect, computer experiments where we define initial conditions and then look at the time evolution of our molecular systems according to Newton's law, force equals mass times acceleration. And with that, and quite amazing advances in both algorithms at our disposal and computing power that we can use, it is now possible to go from molecular simulations of individual proteins, let's say, all the way up to systems that are approaching the scales of small organelles. And here on the right, you see some of our recent studies related to COVID, looking at a patch of a viral membrane decorated with spike proteins. But today, I will look back also and look at ongoing work in our my group and with my collaborators related to the question of molecular processes in autophagy. And I will take you from, as Doro already indicated, from the initiation of the phagophore and the morphology of the ATG9 vesicles to the preparation of the autophagic membrane by phosphorylation of the PI lipids and by lipidation, so the attachment of ADG8, LC3, then to the recruitment of cargo into the expanding phagophore, and at the end sketch some of the work that now then lead to the delivery of the autophagosomes to the lysosome. And this partly is ongoing work that hopefully we will learn more as time evolves. So let me start out with the phagophore initiation. And in case of yeast, there's some really beautiful work from the Osumi lab that indicated that a very small number of ADG9 containing vesicles provide the seed of the growing phagophore. And the number that came out from these imaging experiments was 3. And this is interesting to a membrane biophysicist, because if one fuses 3 vesicles of the same size with each other and preserves the luminal volume and the area of the bilayer, then one has reached an area to volume ratio such that the lowest energy state is actually the phagophore, this cup-shaped structure that you see here at the bottom after the fusion. And this is remarkable, because that would mean 3 is the smallest number to initiate this, and apparently nature has found a solution to this. However, when Amir Bahrami, a postdoc in my group a number of years back, tried to simulate that using, in this case, a molecular simulation model that is slightly coarse-grained, but with explicit lipids and explicit water. The water is not shown in the movie. What he found was quite different. The fusion of three vesicles always led to tubular structures, and that turned out to be also what membrane theory, or the theory of elastic fluid membranes, suggested would be a deep metastable state. And this is indicated here with this energy diagram shown on the top right, that the tubular structure is separated from the disk shape and from the bowl shape by actually two barriers. And in particular, the tube to disk barrier is high for typical membrane elastic coefficients on the order of 40 kT, and it persists even as more and more vesicles are fused into the tube. Only the second barrier disappears. That comes out of work from the Knorr and Limova and Lipovsky. So this was an interesting and somewhat puzzling question, but thanks to work that Doro had already mentioned, work with Jim on the ATG1 complex, we had an idea of what could be a possible solution to this problem. And that was this rather remarkably double crescent or S-shaped ATG17 subcomplex of the ATG1 complex, which has just about, where the arcs have just about the dimensions of to grab the vesicles. And we know from the imaging experiments done in the Hurley lab that there are about 10-ish or so copies of this complex present at the phagophore assembly site. So what Amir Bahrami then set out to do was to look at ATG17 in more detail, and he realized also in collaboration with Jim Hurley's group that there are extensive basic patches on this protein that could serve as membrane interaction sites. So what then Amir did is he built a coarse-grained S-shaped model of the pore and combined it with a membrane fluid elastic model of representing the membrane in as a triangulated surface, but in a fully dynamic way. And so what he then did is he looked at topologies of freshly fused spheres. So they're connected through very thin necks with each other, tethered together by these S-shaped HG17 parts. And here you see one of these dynamic simulations that he ran, and consistently with high yield, what happened was that instead of going to this metastable deep trap of the tubule, the fused spheres quickly shaped into the cup shape of the initiated phagophore. So this was actually very, very encouraging, because additional fusion events, and we showed that explicitly with this cup shape, so further ATG9 vesicles were to be fusing, would actually maintain the cup shape because it is the low energy shape of this system. So, in Jim's lab, then they conducted mutations of these basic patches, and they found that indeed they led to at least a reduction, sometimes a very significant reduction in the autophagic flux. So, but what do these ATG9 vesicles actually look like? And this is the outcome of a collaboration we had with Sasha Martin's lab a few years ago. In our computer models, what we found, this is a model that is based on this very detailed and thorough accounting of the molecular components of the ATG9 vesicles, and then taking the structures and including also the disorder parts, which are quite extensive for these proteins, and place them on the vesicles of about the right size. And so this was work that Sören von Bülow, a PhD student who actually will defend in just a week or so, he's put them together. And here is the model that he obtained. It's a full three-dimensional view of an ATG9 vesicles. And there are important messages from this. First one is that this is actually very densely packed with proteins. And in particular, there are these very large disorder parts that make it rather challenging for the lipidation machinery to even reach the surface of this membrane. And the second interesting observation was that we have, of course, these ATG2 proteins that are quite substantial in size, and that are thought to play an important role in the further elongation steps by facilitating the transfer of lipids from the reservoirs, in particular from the ER membrane. And here is, however, there was one puzzling result at the time when we published this study, that the numbers that had been found or reported for the lipid transfer rates through ATG2 and similar lipid shuttles were very, very low. And it turns out that these numbers actually were based on a misinterpretation of the fluorescence quenching experiments that were conducted to actually measure lipid transfer rates, because ATG2 is like a single file shuttle of lipids. And the consequence of this is, when one looks at this a bit more carefully from a theoretical point of view, is that the rate of lipid shuttling is not the rate of relaxation of the fluorescence intensity. Actually, the rate of relaxation is the shuttling rate divided by the total number of the lipids that are involved in the equilibration process. That is the number of lipids on one and the other vesicle. And of course, even for these small vesicles, that leads to many orders of magnitude difference in relaxation rate and shuttling rate. And with that correction, it is now quite reasonable that a significant fraction of the lipids can actually be delivered by these lipid shuttle proteins. But now moving on in the process of phagophore growth and elongation, we are now at a stage where we have a membrane, but this membrane has to be prepared for receiving cargo. And the first and very important step in this preparation is the phosphorylation of PI lipids by a PI3 kinase class 3 enzyme. And this is a protein that has been studied by many people, and then we had possibility of collaborating on the function of this protein with the Hurley group. And what they were particularly interested in is this the Becklin end of this very large PI3K complex, because Becklin is the part that has the primary interaction with the membrane surface. And there, Becklin appears to be also the site targeted by a protein Rubicon that is associated with autophagy inhibition. That was very interesting, because this interaction was placing a Rubicon helix right above a small beta sheet structure. So where did we come in? We came in by looking in a bit more detail at the interaction of Becklin with lipid bilayers. And again, Sören von Bülow conducted these molecular simulation studies, right, when he started out with his PhD. And what he found was that, as had been expected, the Becklin interacts with this aromatic finger domain very nicely with the membrane, stays very well anchored. However, this interaction could be strengthened if this small beta sheet unfolded, because then the additional aromatic residues that had been pinpointed could actually insert into the membrane, and leading to a much stronger membrane binding interaction. And so we could provide a dynamic and molecularly detailed view of this interaction. And then the speculation or the hypothesis that resulted from this was that Rubicon was the stabilizer of this beta sheet, thereby weakening the membrane interaction. A similar effect happens when HIV-NEF binds, and apparently also can stabilize the beta sheet, whereas a peptide designed in Beth Levine's group, TAT-Beckling-1, that is very similar in composition or nearly identical with this short beta sheet structure, actually appears to be dislodging it and leads to an activated autophagy. So now we have phosphorylated lipids. So we have PI3P on the membrane, and the next step in the development of the phagophore is now we need to lipidate it, which means we have to covalently attach LC3 or ATG8 molecules in a conjugation mechanism that is quite similar to ubiquitin conjugation. So we have an E1, E2, and an E3 system. And it is these LC3 and ATG8 molecules that then provide interaction sites with autophagy receptors for cargo recruitment. So here we collaborated again with the Martens and the Hurley groups to look specifically at the function of ATG16, so the E3-like enzyme in this process. And we were primarily interested, again, in the interaction of this protein, or actually the complex, with the membrane. And we know there are now two interaction possibilities, one in the coil region by helix binding to VP2, which self-binds to the membrane, but then a rather interesting one at the N-terminal end with a direct interaction of the ATG16 complex with the membrane. And so the model that you should have in mind when you, for the following few slides, is this bimodal interaction of ATG16, both on the C-terminal end through VP2 and possibly at the N-terminal end where the catalytic function resides, so where lipidation will then occur at the C-terminal end, at the N-terminal end. So what Shanlin Rao, a postdoc in my group that joined us specifically for the ASAP project, has been doing is to look at these membrane interactions with molecular dynamic simulations. And she used primarily atomistic simulations, but also some coarse grain simulations that I will get to. So in these atomistic simulations, first of VP2D, what she found was that she could place VP2D above a membrane of suitable composition with a small amount of PI3P, and the protein would adhere to the membrane and bind to it. Not only do that in a spontaneous fashion, but it would interact with the membrane through the sites that had been suggested based on biochemical and structural studies, and a site in blade 5 and blade 6, and additional interactions in other parts in the vicinity. But what she also saw, which something that we did not anticipate from the start, was that during the simulations, a rather long loop, the 6CD loop, actually became structured and short helices formed. And these helices also adhered to the lipid bilayer. So this was also the time when AlphaFold became available. So we then started to look at model structures, and lo and behold, what Shandling found was that this very long loop was predicted by AlphaFold to contain two helical structures, albeit with relatively low confidence, but right at the positions that she had seen the helices emerge spontaneously in her simulations. And what's also interesting, of course, is that these helices have amphipathic character with a quite pronounced hydrophobic moment, suggesting that they are membrane binding. And indeed, in long simulations then, again, starting with an unbound BP2D, what Shandling found was that the helices inserted themselves into the bilayer, and in addition to the PI3P interactions, which of course are targeting this protein to the chemically correct membrane, we have these additional membrane interaction stabilizers through the insertion of amphipathic helices. So this is one interaction mode. The second interaction mode has been proposed and identified by Lystad et al. And that is an interaction on the other side of the protein, in the complex ATG12-516 through a short helical motif presented by ATG16. So Shandling also looked at that with atomistic macrodynamic simulations, placing the protein near membrane surface, and found that indeed the complex interacted with membrane, again with a small component of PI3P. And what was interesting, though, was that in these membrane interactions, this rather long helix actually distinctly broke up, and you can see this in the structure. There's a little hinge there, and at this hinge, one of the helices, helix alpha 2, on the left here in this bottom right structure, actually rotated by nearly 180 degrees to present the hydrophobic residues that are in the structure facing the proteins now after rotation towards the membrane. These are the residues Phe32, isoleucine 35, and isoleucine 36 that had been identified also by Lystad et al. As important membrane interaction residues. So while these simulations were running, Leif Jensen in the Hurley group conducted some really beautiful experiments that looked at these membrane interactions in a rather peculiar way, namely by probing the strength of the interactions as a function of the curvature of the membranes. And this is done by pulling nanotubes, membrane nanotubes, using an optical trap out of a GUV, and then looking at the sorting of the protein from the background GUV onto this nanotube in a independence on the tube radius, and therefore independence on the curvature. And what she found was that the ATG12-516 complex exhibited rather strong curvature sorting over this range of up to 0.03 inverse nanometers. Factors of 30, 40, and more were found. And VP2 alone already also exhibited some curvature sorting, and preferring positive curvature, albeit to a somewhat smaller degree. So Shandling then looked at this in more detail. She identified this small alpha 2 helix as the primary membrane interaction site at the N-terminal site, and stably inserted in the membrane in this rotated position, and then use a computational curvature sensing assay that we have developed a number of years back, and I will describe in more detail in slides that come in a couple of minutes. But in essence, we have a sinusoidally warped membrane, and then we look at the distribution of a bound protein in a long simulation um, with respect to the preference for different regions and therefore different curvatures of the membrane. And what Shandling found, as you can see in the histograms in the bottom right, is that this alpha-2 helix, despite its really short length, has very pronounced curvature preference in her simulations, pushing it towards a preferred curvature of about 0.025 inverse nanometer, so about 40 nanometer radius. And so this again turns out to be consistent with the experiments, because as Leif Jensen could show, is mutations of these key residues on that helix abolished the curvature sensitivity endowed by the N-terminal. But VP2D itself is also curvature sensitive, and you may have guessed it already. The primary drivers of that are indeed these short alpha helices of amphipathic character in the 6CD loop. And this is illustrated here in, again, the distribution of sampled curvature by these helical regions in the reddish color, compared to the distribution that we have randomly across this sinusoidally warped membrane. So we have indeed two elements of membrane binding reconstituted in our computer. At the C-terminal side, through PP2D, at the N-terminal side of ATG16 through this small helix, and both of them are curvature sensitive. And the small helix on the N-terminal end actually is the primary curvature sensor, which at least at the qualitative level is consistent with the experiments by Leif Jensen. So now we have the LC3 EDG8 on the membrane, and the membrane is thus ready to receive cargo. So how is cargo delivered? And this brings me to a project that was carried out by Ram Bhaskara when he was a postdoc. He's now running his own independent group here in Frankfurt. So Ram started a very intense collaboration with Ivan Dikic and his group on FAM134B, which Ivan Dikic's group had previously identified as an ER-phagy receptor, so a receptor that is used to target ER and particularly also misfolded proteins like collagen in the lumen of the ER towards the autophagic machinery. So when Ram started out, we had very little information on the structure of this protein. And of course, as those of you familiar with molecular dynamics simulations know, if you don't have a structure, it's very difficult to start a simulation, because then you rely on modeling. But this was actually an interesting modeling target, because we knew quite a bit about the topology of the reticulin homology domain, which is shared with other reticulin-like proteins, and that we have two transmembrane helical hairpins, and as Ram could show, distinct and functionally important amphipathic helices connecting these transmembrane helical hairpins. So Ram built structural models and then relaxed them with very extensive MD simulations, both coarse-grained and at the atomistic level, and then developed a functional assay for membrane curvature induction, because Dikic's lab and their collaborators have previously shown that FAM is a very powerful inducer of membrane curvature. And as you can see in this little movie on this side, this is something that we can see very clearly also in molecular simulations. Here, the insertion of a FAM protein into a lipid bicell drove the bicell into a vesicular state, and we could use this assay to identify the main drivers of of membrane curvature induction in FAM, and turns out it's not so much the transmembrane, the wedged transmembrane helical hairpins, as it is these distinct amphipathic helices that drive curvature induction. And then by teaming up with Misha Kudryashev's lab here, when he was still here in Frankfurt, actually at my institute here, we could recapitulate this by EM imaging using quite a large number of constructs with various parts deleted, and indeed could see that FAM 134B RHD drives very strong curvature. We observed vesicles of diameters in the sub 50 nanometer range induced by this, and it is indeed the amphipathic helices that are the primary driver of this transition. So this is interesting and brings me back to this curvature induction versus curvature sensing point and the assay that we have been using to study this. And here you now can see the assay of curvature preference probing in action by looking for reference at the small helix, transmembrane helix, and comparing it to FAM 134B RHD. And what you can see is the FAM protein on the right actually pulls the warp to it rather than diffusing into the peak of the membrane, and then just stays there bound. And that leads to this remarkably strong curvature preference, as indicated by the sampled curvature distribution at the site of the protein shown in green, and compared that to the lipids and the calp protein, which has no curvature preference, in red and in blue. So we have a very strong curvature preference, and that actually combined with the observation that FAM 134B in the ER forms distinct punctate structures, raised the question whether clusters now of this protein would amplify this effect. And that's indeed what we found in large computational studies by placing multiple FAM proteins in the membrane and then running molecular dynamic simulations. What we found was that these proteins, they diffuse around until they find each other. And once you have reached a critical size of, in this case, 4 to 5 FAM proteins co-localizing in the narrow, small patch of membrane, they could, with a bit of help, induce spontaneous budding of the membrane. So what was this bit of help? This was a computational trick that we developed to overcome the problem that it is very difficult to make topological changes of membranes under protein boundary conditions. So what we did is we created a bit of asymmetry between the upper and the lower leaflet of the membrane by moving some lipids between them. And this asymmetry, in effect, just modulates the kinetic barrier for this budding transition. So the critical point here is that each FAM protein induces local curvature. Clusters of FAM proteins have an additive effect, and when they co-localize and actually also interact with their amphipathic helices, they are really very powerful membrane shapers. So the picture that then emerged for this process of Autophagy, where you have to remove both ER membrane and ER luminal content, such as misfolded collagen, is that the FAM proteins co-localize with the target, their unfolded regions, and with their LIR motifs interact with the LC3 proteins on the phagophore, and this co-localization, the formation of punctate structures, deforms the membrane and facilitates, maybe with a bit of mechanical help and maybe other factors that are not yet fully characterized, then the removal of the ER. So now we have a substrate on our phagophore but then it has to be still delivered. And as it turns out, this is an area that I have not quite done so much yet in terms of modeling, but we have looked at some interesting aspects, and we have ongoing work that also really completes the life cycle of the phagophore, hopefully in not too distant future. So what we have looked at, this is again in collaboration with the DKICH lab, is the action of the ATG4 protease and its modulation by phosphorylation. So the ATG4 protease is interesting in that it functions both early in autophagy by processing the pro-LC3 protein through proteolytic cleavage of the C-terminal tail, which activates it for then subsequent lipidation reaction that I showed to you. And but it also is the protease that eventually removes the LC3 from the autophagic membrane and thereby enables the fusion of the mature autophagosomes with the lysosome or the vacuole in yeast. So what Ram Bhaskara looked at was the effect of phosphorylation on LC3 on the action of ATG4. And the reason that he looked at that was findings by Lina Herhaus in Ivan Dikich's lab that LC3 phosphorylation is conducted by TBK1, so PENK binding kinase 1 at 2 positions, and that this phosphorylation prevents premature cleavage of the LC3 from the autophagosome by the ATG4 protease, and thereby ensures autophagic flux, and in this experiment is sustained mitophagy. So how does this come about? And again, molecular dynamic simulations can give us an understanding of the effects of small chemical modifications here, the phosphorylation of 2 amino acids. So here you see the complex of LC3 bound to ATG4 run by molecular dynamic simulations, and there's nothing special happening here. It actually is nice and stable because the 2 serines are unphosphorylated. So it forms a nice complex, and making this LC3 in the unphosphorylated form a good target for the action of the protease. Now, when then Ram looked at the effect of phosphorylation, he ran the same simulations now with phosphorylated 93 and 96 serines, and what he saw was that the complex fell apart really rather quickly. In particular, the region involving the 2 serines completely opened up, and meaning that the interaction now of LC3 in the phosphorylated form with ATG4 is very, very unstable. And so this explains how phosphorylation prevents ATG4 action on LC3. But there's another interesting aspect to this, that is what Ram in the simulations of LC3 alone in phosphorylated and unphosphorylated forms also saw was there is an additional allosteric-like effect in that in the phosphorylated form, the C-terminal tail, which has to be cleaved, is actually interacting strongly with the phosphoserine, thereby with phosphorylation prevents also action on the cleavage reaction. So as I said, we haven't quite made it to modeling the delivery of the cargo into the lysosome, but we are on our way. This is results of a really amazing experimental study conducted by Florian Wilfling and his collaborators in Martinsried. And we have started to conduct initial modeling on this process, looking in particular also at the phagophore vacuole interaction. But I guess you will see more on this in Florian's talk in at the end of summer or and actually September 27. So to conclude, I just want to show autophagy is not all we do. I'm showing this also to give you an idea of that molecular simulations has come quite a ways over the last few decades. One project that I'm really truly excited about is molecular dynamic simulation studies of intact nuclear pore complexes in collaboration with the lab of Martin Beck here at our Max Planck Institute. And this is interesting for many reasons, complexity, but also size. As you can see here, this is a large complex by any measures when you compare it in particular with a ribosome. So what Mark Sigel, who I already mentioned for his work on membrane remodeling, has been able to do is set up dynamic simulations of this system in periodic box with a double membrane envelope structure, and look at this various questions in mind. But in particular, we were interested in the relaxation of the nuclear pore in response to mechanical cues, in particular tension, mechanical tension in the double membrane nuclear envelope, because what our experimental collaborators had observed was that the pore can indeed undergo opening or constriction and dilation dynamics. And this is something that we could actually replicate to some degree already in our computer simulations. Another area that I just want to show very briefly, I'm also extremely excited about is pore formation in membranes. And in this case, these are pyroptotic pores created by gasdermin D in inflammation, particularly in response to infection. And we can simulate these pores both at the molecular scale when look at ion passage for tiny, small oligomers of gasdermin, and for larger oligomers. And here you can see a top-down view where you can see the spontaneous opening of the pore through lipid efflux from the arc of the gasdermins, and then the formation of the ring-shaped structures. The arcs are large, and these slits-shaped structures seen in the AFM experiments done in Daniel Müller's lab. So I hope I've given you an idea of what simulations can do, and in particular also with such complex processes such as autophagy, that we can zoom in on key molecular events all the way along the autophagy pathway and connect molecular factors such as post-translational modifications, phosphorylations to specific functions. And the last two slides I showed to also give you an idea that looking forward, we really can think of conducting atomically, at least molecularly resolved molecular simulation studies, not just of isolated molecules, but really of larger assemblies all the way to small organelle structures. With that, I stop, and I want to thank my very many collaborators, and I think I mentioned the experimental collaborators already, but I want to thank in particular the members of my group who have conducted these studies and have really delved deep and been as a result able to educate me and excite me, and I hope I could also share some of those insights with you, and hopefully you find this at least interesting. And I will stop here. Thank you, and we'll try to answer questions you might have. Thanks.
Thank you, Gerhard. That was not just interesting, that was just absolutely fascinating. I think I'm speaking on behalf of the whole audience that everyone's just mesmerized by what you just showed us. I see that we have already one raised hand. Maybe we can get, yeah, there she is. So Ayush, I think you should be able to speak and ask your question.
Right. Hello. Am I audible?
Yes.
So I wish to inquire about, has there been any sort of investigation into how or rather why the mechanism for the preference that ATG 12.5.16 Li has for the positive curvature, or VP2B has for the positive curvature? Is there any sort of?
Yes, thank you. This is an excellent question that I did not touch upon. So the question is, we saw this with an experiment and a simulation, distinct preference for positive membrane curvature for ATG 16. And so why is that? So of course, this is to some degree an open question, but the possible explanation is localization on the growing phagophore. So you can think about where would you want to have the strongest action in terms of lipidation, and so this would indicate this would be at the rim of the growing phagophore, where of course the curvature is rather large. So that's the I think the likeliest explanation for this preference. And there are some imaging data on plant systems that would be consistent with this. But this is something that we still maybe others, Jim and others, might have other opinions on that. But for me, it's the primary driver would be to have proper localization. The second possibility, of course, could also be an element of targeting, so to amplify the effective targeting ATG 16 to the right kind of membranes.
Thank you, Gerhard. I see we have another question from the audience. Radhika Kara, if you're still there, I think you should be able to ask your question in person as well now. Okay, I see she removed her raised hand. Perhaps that was a mistake, or she's not able to speak at the moment. So if people still have questions, I think we have time left for one or two final questions. What I may do is quickly squeeze in a personal question, because I had a few myself, Gerhard. I was wondering, for the FAM134B, the autophagy receptor, it's quite interesting that you need 4 or 5 proteins to come together to induce a curvature. Did you find any evidence that when these soluble domains come together, that they then would sort of reposition the alpha helix to insert in the membrane and sort of communicate that we are together, this is the moment to sort of make this vesicle-like structure?
Yeah, so this is an interesting question, whether as the clusters, so this kind of course, the initial collision is rather unstructured. So when they find each other, and then once they hit each other, they actually do seem to adopt preferential interactions with each other. So what we have seen in our simulations is almost pyramidal arrangements with contacting amphipathic helices, and the amphipathic helix is also forming the rims of the pyramids. And what is interesting is if one looks at the AlphaFold prediction for FAM, it's mostly just helices. You have the hairpins are definitely picked up, and then you have a long helical regions, but you also see actually some indications for possible helix contact. So there might actually be something to these observations, that there is structure emerging. But of course, I have to caution that the representation that we use there has still many flaws, both in terms of its energetics and also in the time scales of these processes. So I think there are these superstructures. I would be rather careful. I would use them more as maybe inspiration for targeted experiments rather than as genuine models of higher order structured assemblies. But they're definitely suggestive and interesting.
And then I think I can leave the last question of the season for Doro.
Yeah, Gerhard, I was just actually thinking now, it's more of a methodological question. So in your simulations, you usually have a naked membrane with at most appropriate composition for as much as it's known, and then one or two active membranes that, proteins that you are looking at. So usually though membranes do have other proteins that can also be transmembrane or peripheral. And I was wondering whether there is any method developed that can account for the role of these proteins that as a space fillers, or because sometimes they don't really have activity on these membranes and for the one process, but they are still there. And so whether this has been ever addressed.
Yeah, Dora, this is an excellent question, of course, that many of us have been thinking about. And we have, of course, the same question in modeling cytosolic processes. And there has been, in recent years, increasingly this effort to include a more realistic representation of the cytosol, which, of course, is itself densely packed with proteins and other biomolecules. And this is even more true of nearly all the membrane systems. If one looks at the copy numbers of proteins in membranes, I mean, these are, for the most classic case is mitochondrial membrane, is barely a lipid bilayer. It's mostly proteins with a sprinkle of lipids. And other membranes, of course, are a bit more dilute. So clearly, I think as we are moving forward, this will become an important topic of research to understand whether these are just these other proteins and of course the modifications are just passive bystanders and they are essentially inert, or whether they change the physicochemical environment enough that they would have to be included, maybe also in of course, in reconstitution experiments, not just in simulation. So this would be, I think, again, excellent question, and unfortunately, no clear answer. But from the developments on the soluble side, I would say I wouldn't be surprised if there is additional interesting complexity coming from these interactions.
Thank you.
All right, then thank you again, Gerhard, for this fantastic presentation. And all that's left to do is invite everyone for our next season, which is here on the screen. If you haven't registered yet, you can still do it. And otherwise, we look forward to see you after summer. I wish everyone a very nice summer break. So thank you.
Thanks, Gerhard.
Thank you, everyone.
It was great.
Bye. Thank you all.
Thank you.